Reproductive system development is the series of events that an embryo goes through to sexually differentiate into a male or female, with regard to the gonads, genital ducts, and external genitalia. The process starts at conception, when the gametes, the sperm and oocyte, fuse into a single cell that has either XX sex chromosomes in a female or XY sex chromosomes in a male, establishing the genetic sex of the embryo. Through the first 5 weeks of development, though, sexual development is basically identical for both sexes. At that point, the embryo is made up of 3 primitive germ layers: the ectoderm, the mesoderm, and the endoderm. The mesoderm also has 3 parts: the paraxial mesoderm, flanking the embryo's future vertebral column, the intermediate mesoderm, which is just lateral to that, and the lateral plate mesoderm, which is the most lateral of all. The intermediate mesoderm on both sides of the embryo condenses into 2 cylindrical structures called the urogenital ridges. Each urogenital ridge runs parallel to the embryo's future vertebral column, and organizes into a cylinder of mesoderm called the nephrogenic cord. Most of the nephrogenic cord goes on to form urinary structures, but a strip of it in the middle gives rise to the gonads in males and females. This portion that gives rise to the gonads is called the genital or sometimes gonadal ridge. This genital ridge has a mesoderm core and is covered with epithelium. Gonad development, interestingly enough, starts in a tissue outside the embryo called the yolk sac, which is lined with endoderm cells, and connects to the embryo through the vitelline duct. Early on in development, some endoderm cells from the wall of the yolk sac differentiate into primordial germ cells, and they start to migrate, meaning that they physically move along the vitelline duct, eventually to the primitive digestive tract, and finally to the dorsal mesentery, which is a sheet of tissue that anchors the digestive tract to the posterior body wall. From there, the primordial germ cells march along the dorsal mesentery until they get to the genital ridge, arriving there around week 6. And those primordial germ cells settle in the epithelium. Once there, they send out chemical signals to the cells in the genital ridge, which makes them self-organize into an undifferentiated gonad, which can still develop into either testes or ovaries. The epithelial layer of the gonad then forms primitive sex cords, which are epithelial projections that penetrate inwards into the mesoderm layer of the gonad. Around week 7, sex chromosomes start expressing genes that determine gonadal differentiation. In a male, genes in the sex-determining region of the Y chromosome, or SRY for short, code for a protein called testis-determining factor, which initiates the development of the testes. The primitive sex cords mature into medullary cords that grow longer and carry the primitive germ cells deeper into the mesoderm. The surface epithelial layer of each gonad thins out to become the tunica albuginea. Later on, the medullary cords develop into 3 ductal structures inside the testes: the seminiferous tubules, straight tubules, and the reedy testis. Of the 3, the primordial germ cells settle down in the seminiferous tubules, and that's where they mature into spermatogonia, and lay dormant for a number of years. During puberty, the spermatogonia wake up and start dividing over and over again to give rise to sperm, which are the male gametes. During week 8, some cells in the walls of the seminiferous tubules differentiate into Sertoli cells. Sertoli cells surround the primordial germ cells and secrete anti-Müllerian hormone. In addition, cells between the seminiferous tubules differentiate into Leydig cells, and they secrete testosterone. Anti-Müllerian hormone and testosterone masculinize the rest of the male reproductive tract. Now, in a female, since there's no Y chromosome to secrete testis-determining factor, the undifferentiated gonads develop into ovaries. In this case, the primitive sex cords also extend toward the center of the gonad, but they degenerate soon after. The surface epithelium proliferates again, and forms a second set of projections called cortical cords. The cortical cords rearrange to form nests of follicular cells that surround the primordial germ cells. A primordial germ cell and its ring of follicular cells make up a primordial ovarian follicle. And inside it, the primordial germ cell differentiates into an immature oocyte during fetal life. The immature oocytes are halted in the first prophase of meiosis I until puberty, at which point they turn into secondary oocytes, which are the female gametes. The rest of the reproductive tract acquires female characteristics in the absence of testosterone. Now, the differentiation of the gonads leads to the phenotypic differentiation of the genital ducts and the external genitalia. The genital ducts are initially undifferentiated tubular structures that run down the embryo's back inside the two nephrogenic cords on either side of the embryo. The first is the mesonephric or Wolffian duct, which gives rise to the male reproductive duct system. The second is the paramesonephric or Mullerian duct, which is lateral to the mesonephric duct and gives rise to the female reproductive duct system. These ducts start in the thoracic and upper lumbar region, and continue down the embryo's back until they open into the part of the cloaca called the urogenital sinus, which gives rise to the urinary tract structures in the external genitalia of both sexes. In males, Mullerian inhibiting factor makes the paramesonephric or Mullerian ducts degenerate into two vestigial bodies called the appendix testis. The mesonephric ducts, on the other hand, grow long under the influence of testosterone, and form the reproductive ducts outside the testis. The efferent ductules look kind of like the teeth of a comb, and they connect the intratesticular straight tubules to the epididymis. The epididymis is where sperm go to mature, after they've been produced in the testes. After the epididymis, there's the vas deferens, through which sperm will be released during ejaculation. An accessory gland called the seminal gland develops from each vas deferens, and it secretes a nourishing fluid for the traveling sperm. At the end of each vas deferens, below the opening of the seminal glands, there's an ejaculatory duct, which opens into the urogenital sinus, which forms the male urethra. In females, on the other hand, the ovaries don't make testosterone, so the Wolffian ducts degenerate. Also, the ovaries don't make Mullerian inhibiting factor either, so the paramesonephric ducts persist. In the upper regions, the two paramesonephric ducts form the fallopian tubes. In their lower regions, the ducts fuse and form the uterovaginal primordium, which is a tubular structure from which the uterus, the cervix, and the upper third of the vagina develop. The lower two-thirds of the vagina develops from the urogenital sinus, when the external sex organs differentiate. The external sex organs, though, also go through an undifferentiated stage, during which the lowermost portion of the urogenital sinus opens up in the genital region. Flanking the opening on either side, there are two sets of symmetrical folds, the urethral folds and the labioscrotal swellings. Above the opening, there's a small bit of tissue called the genital tubercle, which first turns into a primordial phallus for both sexes. The differentiation of the external genitalia into male external sex organs starts when testosterone levels start to rise. An enzyme called 5-alpha reductase, which is found in a lot of tissues in the body, including the skin of the external genitals, converts testosterone into dihydrotestosterone, which is its more potent form. Starting with week 9, dihydrotestosterone masculinizes the external sex organs of male embryos. The urethral folds fuse on the midline and form the penile urethra. The primordial phallus grows longer, and its tip forms the glans penis, while its body gives rise to the body of the penis, which encloses the penile urethra. The labioscrotal swellings fuse as well, giving rise to the scrotum. For genetically female embryos, in the absence of testosterone, the urethral folds stay unfused and form the labia minora. The labioscrotal swellings form the labia majora, and only fuse in their anterior portion to give rise to the mons pubis. And finally, the primordial phallus shrinks and turns into the clitoris. All in all, phenotypical differentiation is done around week 12, so this is about as early as you can tell the sex of an unborn baby. Finally, there's one more step in the development of the reproductive system. The gonads have to descend to their adult positions, in both males and females. Starting with males, the testes initially develop near the anterior abdominal wall, but they're anchored by the gubernaculum, which is a fibrous cord that attaches to the bottom of the scrotum, and also has a really awesome name. The gubernaculum starts to shorten, pulling the testes down towards the scrotum through the inguinal canal. The inguinal canal forms when a projection of the peritoneum called the processus vaginalis herniates through the abdominal body wall. By week 12, the testes reach the inguinal canal and move towards the scrotum. On their way, they pull the vas deferens as well as the testicular artery and vein with them through the inguinal canal. These structures eventually form the spermatic cord. This journey is usually over by the last month of pregnancy, and the abdominal muscle layers that came along form the layers of the scrotum. Now, in female embryos, there's also a gubernaculum that attaches the inferior part of the ovary to the future labia majora, and it shortens to pull the ovaries down the same way it would if they were testes. When the uterus forms, though, the middle of the gubernaculum attaches to it and separates into a superior portion called the cranial genital ligament, which becomes the ovarian ligament and anchors the ovaries slightly above and on either side of the uterus. The inferior portion, called the caudal genital ligament, becomes the round ligament, connecting the uterus to the labia majora. Alright, as a quick recap, the reproductive system goes through an undifferentiated phase of development, which is the same for both sexes. But in week 7, the primitive structures differentiate into male or female sex organs, according to the sex chromosomes that the baby's had since conception. First, the gonads develop into ovaries or testicles, and the differentiation of the gonad acts on the rest of the reproductive tract, which gives rise to the corresponding male or female sex organs. Both the ovaries and the testes initially develop in the abdomen, but descend during pregnancy to reach the pelvic cavity or the scrotum.